Okay, because it is going to be long, I think I'll go ahead and start now. So I'd like to welcome you all to the second in this introduction to the new Bacterial and Viral Bioinformatics Resource Center. This center is hosted jointly by the University of Chicago, Argonne National Labs, the J. Craig Venter Institute, the University of Virginia, and the Fellowship for the Interpretation of Genomes. And of course, it's funded by the National Institutes of Allergy and Infectious Disease. Today, we're going to be talking about bacterial data in the new BV-BRC and what changes people who are coming from Patrick might see. So I just wanted you to be aware that we have some upcoming. We've had one webinar and there are some that are upcoming. Next week will be the viral data. So I would encourage you to attend that, especially if you are a Viper or IRD user, or just actually anyone who's interested in viruses. But we have these, the dates of these posted on the website for you to look at and join. I also wanted to make you aware that we have another webinar series that we're about to launch very soon. This is a cross BRC webinar series with the people from ViewPathDB, which are vectors and eukaryotic pathogens, and the BV-BRC, which are bacterial and viral pathogens, or in all bacteria. And so you may be thinking, I don't work with ticks, why would I want to come to this? Well, we'll be showcasing a lot of different tools and abilities in these webinars. So if you're just interested in seeing these things, you should register to see that. And to register, you would go to the ViewPathDB and you can register for it there. It's all totally free. So as we, the BV-BRC resource website is new. We're in the process of developing it. And we'd like to hear from you, your comments, your concerns, things that are blocking you, because we want you to use the resource. We want you to feel comfortable using it. And when you're having problems, we want you to tell us. If you just get frustrated and walk away, we don't know. We think everything's perfect. And you know what? We know it's not perfect right now. So we want your help. So how do you contact us? You, on the top of every page in the BV-BRC is a help tab. You click on that, that opens a drop-down box. You would click on the contact us. That opens a pop-up window. You describe your likes, your dislikes, where you were blocked, your happiness, your unhappiness. We want to hear it all. So you enter that in the text box and then you hit submit, and that will come to us and we'll start working on addressing that issue. So enough of the background stuff. What I presume you're here today about is to find out about the bacterial data that's in the BV-BRC. We have pathogen data, and actually we have other bacterial data as well, and I'll show you those and how to find them. Now, one of the things, usually when I do these kinds of things, I start at a high level, like the taxon level, and work my way down, because we're asking you to embrace our new combined resource. I decided I could be a little uncomfortable too. So I'm starting at what would normally be the bottom for me, at individual genes, and working my way up. So I'll be showing you all the information associated with the gene or protein page, and then going on to the genome, and then going on to a different taxonomic level. And then we'll get to private data. And in all of this, I will be describing the pages and the tabs. So let's get started. You need to go to bv-brc.org, and I'm going to show you what it looks like. So here we go. Buckle up. All right. So let me put that away. Here's the page. Let me make it nice and big so everybody can see this. If you were on the last series, or the last lecture, it was all about the landing page and all the tabs. Today it's going to be about all the data. But like I said, we're going to start with an individual gene. So let's imagine that this is my favorite gene in the world, and I've been working on this for most of my life. And when I'm at GenBank, I can see all the information associated with this gene. And I have links to the protein and the region and the CDS, and I have the FASTA structure, and I can see, or the FASTA sequences, sorry, and there are certain hyperlinks and things like that. So if I'm trying to see if I'm interested in this new site, what does it have about my individual gene? So let's go look there. And the way I would do it, rather than take the name, I'm going to take the locus tag, the accession number. Oops, drop me out of that. And I go to the page, and let's pretend I know nothing about this, but I guess I can see that there's a search box here, and I can just put that accession number in there. And let's pretend that we don't know about drilling into data types, and we don't know about adjusting stringencies of search, and we don't even know to push this little search icon. So I just hit return, and pretty quickly it comes up and it's summarizing all the data that we have in the resource for that particular accession number. And here's a summary up here. It knows that I'm looking at proteins, and here are the proteins here, and you see that there are 2 of them. Well, let's click on this. Why are there 2? And when it comes here, it's just showing me 1 because it's automatically filtered on the annotation provided by the BV-BRC. If I got rid of that, then you'd see what it's called in RefSeq and what it's called at the BV-BRC. So if I click on this, oh, I've got to move this over here because you guys are interrupting my view here. You can see that here's the information about this gene here. And when I click on it, it automatically populated this vertical green bar with several downstream functions that you can do once you've clicked on something in the table. I could go to the feature page. I could go to the genome page. I could get the FASTA data. I could see what IDs are associated with this, or I could add it to a group. And you can also see that there's information beyond the bar that summarizes even more data about this, including when it was inserted and things like that. So you might think, well, okay, but I kind of like that GenBank format, still not sold. Why do I want to come to here? Well, let's click on the feature tab. And you can see that it has loaded what we call the landing page for this particular gene or protein. Now, here it's telling me where I am and who it is with the RefSeq locus tag and the BV-BRC identifier, gene identifier for this. And it's also got a breadcrumb across the top to tell you where you are. And I can see when questions come up, well, I'll go ahead and look. Yes, a question is, can we get a recording at the end of today's webinar? We'll probably post it probably by tomorrow. What I'll try to do is remember to, oh, we need a list of these users. Well, it'll be on, you would go into the help and then webinars, and you'll see it there, but I'll tweet something that says that it's available if that helps. Okay, back to the story about this really fascinating protein, which it is a fascinating protein and a very important one in the tuberculosis disease. So you notice that we've got a lot of information here, but there are all these different tabs too across the top, because there's so much information here that we can't put it all on a single page. You'll notice that this shows me the gene neighborhood. I would welcome you to scroll down here and see all the information about this gene. Anything that's blue, and when you mouse over it, That's a hyperlink that will take you to places outside of BV-BRC, so you can see that information. We also have external tools associated with this gene. We can't put everything in here, so I would encourage you to click on that. And I can see that there are more chat things, so I'm hoping that somebody from the team is monitoring those and answering those so I can keep drilling through this. So we talk about the protein families, and this is one place where you can see any genome that is annotated in the resource, be it public or be it private genome, in the annotation, which we'll talk about on April 1st, but you would go to the annotation service. Protein families are assigned to the bacterial and archaeal genomes that are based, Kamer-based, and there are the genus-specific and the cross-genus protein families. But you can also see how many proteins in the entire resource have this particular gene, or this identical sequence, or the identical nucleotide sequence. And it's also showing any genome that is annotated in the resource is blasted against specialty gene databases for virulence factors, antimicrobial resistance genes, essential genes, drug targets, transporters, and human homologs. And so this is summarizing where this particular gene had hits, and you can see it had very good subject and query coverage and percent identity and a strong E-value to these different resources. And then at the bottom, it shows you all the identifiers from different resources that map to those genes, which you could use to search in the resource to find that. If this is where you were coming from, you could insert that into the global search button, which notice it's moved up here into the header once you move off the top page, and it should bring you directly back to where we are now. But but wait, that's not all. There's more information that you see with these tabs. I can look at the genome browser. So I click on that, and this will be one of the few places in the resource where you have a head-to-head comparison of the GenBank or RefSeq information and the BV-BRC information. And you can drill deeply down on this page until you get to the 6-frame translation. So this is a good, if you have a gene that's broken, this is a good place to look here and see what's breaking it. This is another thing we have only at the feature level. And this is one of the main reasons I wanted to start here to show you this. Any gene on any genome annotated in the resource, be they public or private. And let me point out that we have more than 500,000 bacterial genomes in BV-BRC and millions of viral genomes. But for the bacterial genomes, every one of them, we have this function, which is the compare region viewer. And it's looking not only at the particular gene, but the gene neighborhood. And it's looking across all those genomes and saying how conserved is, what other genomes have this particular neighborhood. So right now you can see that you could adjust the size, the number of genomes, you could change the flavor of the protein families, and I'll show you this part in a second. But if I mouse over these, you can see what the gene is called. And if it's the same color, it indicates that it's the same protein family. And you can see here that this particular one in this genome, it's actually broken. This gene is the same gene, but it's broken here. And actually, this is well known in the Mycobacterium bovis strains. This is who we came in on, this red arrow, and you're probably wondering, what is this square box underneath it? I don't understand that. And look, the colors are going down, down, down. This is an indication of the blast hit. For this gene, when it's blasted against my genome, it's a perfect hit. So it's a strong, vibrant red color. but as you get less, as it's less conserved, the color gets less. And then you notice here that this is a small little box here compared to this. That means that this gene, only part of it hit here, and it was not as good of a hit. So that's the color. So right now we have, the default is the reference and representative genomes. This is a group of genomes assigned by GenBank, and those are the gold and platinum standard genomes, ones that people use a lot, have experimented on a lot, that have a lot of data associated with them, or might be one of the few representatives of that particular taxon. So those are all included here, but I could open it up and say, I want to see all the public genomes and look at that, and then suddenly it changes. And you can see the data that's more specific, because now I'm looking at a bigger set, so it's going more into the tuberculosis. And you'll notice something weird, like what's happening here. This is an indication to me that this genome is in multiple contigs. And when it looked for the neighborhood, it could only find part of it here, so it's showing it here. Let's go on to transcriptomics. Several years ago, we went into we got a bunch of microarray data and for specific genomes. And so we have the expression values for that data across for the genomes that are applicable across our database. And this particular genome, H37RV, has a considerable amount of data associated with it. And these are the expression values for this particular gene. So you could look at the log ratio or z-score where it's going up and down. You could look at the metadata associated at the strain level, at the gene modification level, or at the experimental control. And let's say that you were interested in just a single experiment. And I have it here. I'm trying to find what I had said. Here it is. So you know that I don't do this kind of experimental analysis, but let's pretend that I did, and I'm really interested in that particular experiments that are dealing with this. So I could filter on that by putting it in the keyword and then clicking the filter, and I could see where this gene is going up or down in those experiments. And I could also look at this table and then could see the actual values of where this is changing. So really, how cool is that? I just really like this feature. So I can see at a gene level where my gene is going up and down. So if you've done that, so you know your gene's going up and down, you know when it's expressed, and when it's upregulated or downregulated. You might also be interested in things that it's interacting with. So you could click on the Interactions tab, and you can see there's a list up here. These are the genes that have been shown to interact with this particular gene. I could click on the filters here, and I could drill down. This is a good way. Right now, we're looking at one gene. Then we're going to look at a genome. Then we're going to look at a taxon level. And every step up that we take, the data is going to get bigger and bigger. So you're going to need to use these filters to find the data that you're interested in. So we've got some experimental data here. I could say I only want to see TN-seq if I wanted to. I could click on it, or if I change my mind, just click it off. And I could click all of these. And then up here, see this graph icon? I could click on that, and then it gives me a nice little graphic showing me my protein. So you can mouse over each of these and see that information, and the connectors too, to tell you what the association is, what the interaction type is. Let's go on to domains and motifs. Now this, for people who are coming from Patrick, this is new. And I've done domain research before in a bioinformatics sense for some collaborators. We didn't have this in the resource before, but when Viper and IRD, when we joined with them, this is something they've always had. So this is new, and for people who are looking at these things, this is very cool and a brand new functionality that you didn't have before. So this is showing me on this one gene, how many domains are associated with it and where the evidence is from. And if you want to see more information, you can click on the filters and that'll show you different things about it. You could also click on one, and once again, it populates the vertical green bar with possible downstream functions, but it also gives you more information about this particular domain. And then protein structures. We had this for a while in Patrick, and then it went away, but Viper and IRD have always had it. And so now it's back by popular demand. So this is something else that is a good thing that resulted from this marriage between the resources. So this is showing all the different protein structures that are available for just this particular gene. So I can click on one, and this time there's a brand, of course, beyond the green bar, there's information about it, but you can also click on the structure icon. So if I click on that, it's going to open up this structure. And I mean, I don't work with structures, but I enjoy playing with this. And I can also click on different regions in the sequence, and it'll show me that. So, I mean, for me, that's pretty fun. Okay. So this is all the tabs that we started with looking at the data available in GenBank. And I just wanted to show you all the information associated with this same gene in the BV-BRC. So this is gene information. Let's look at a genome and see what kind of information we have here. So I'll click on, I'm going to click on the breadcrumb because I'm going to march my way up the story. And so this will take me to the Mycobacterium tuberculosis H37RV genome. And I can see here that it's one of the reference genomes that's been assigned by NCBI. And yet, once again, here we are at another page. You've got a ton of information on this page, including, I didn't mention this before, the PubMed articles. Anytime you draw on one of the pages at the level, yes, it will launch a search at PubMed and on the fly show you the most recent articles that deal with this organism. Oh, it says my internet. Okay, here we go. You notice we also have a bunch of different tabs associated with this particular genome. So let's start marching through those. This is the overview tab, and you can scroll down, down, down. There's a ton of information here that I encourage you to explore. We have genomic features. We have the protein features. Like I'll keep saying it again and again, any genome, private or public, that is annotated in the resource. We look for EC numbers. We look for Go assignments. We assign them to pathways and subsystems. We give them protein family IDs. We blast them against specialty gene databases, like I described before for that individual gene. So there's all that information here, and you can actually jump to those just by clicking on any of those. And you'll notice here it's showing you in the BV-BRC version and in the RefSeq version what the difference is in, for example, the hypothetical proteins, how many of those are annotated in each of those. So let's continue to march up. I'm going to talk about Amr phenotypes in the next as when we step up again. But I wanted to show you phylogeny. Sometimes you just don't know where a genome exists in the tree, in the larger tree. So several years ago, we generated trees at the order level. There are thousands and thousands of Mycobacterium tuberculosis genomes. We'll be able to see that in a second. This is at the order level up here, and we chose, at the time we generated these trees, 375 high quality genomes. And so if you wanted to see where this strain was, you could do a control find and look for it on the tree. Let me also point out that you can, if you're interested in this, you could click on that and it will select all of those guys, and you could create a group from them, or you could see what are the unique identifiers. You can see here that the name is not necessarily unique, but the genome ID is. So you could do something like that. Now, if you have brought in your private genome, it's not going to be on this tree. And in fact, most of the Mycobacterium tuberculosis genomes are not going to be on this tree, but they're just a subset of this. This is just to give you an indication of where you are in the great bacterial world within this order. At the genome level, we also have the genome browser. So let's click on that. And this, once again, is showing you differences between Patrick, or similarities too, between it's hard for me too. I make the mistake myself. BV-BRC and RefSeq for GenBank annotation. And once more, I can drill down in and make direct comparisons between what this is called in BV-BRC and what it is called at GenBank. So you can see that kind of information. And were this in multiple contigs, I'd be able to click here and go to the different contigs. We provide a circular viewer. In fact, many of the papers that cite us when they're talking about a genome, they will bring this circular viewer in and take that as export it as an SVG and use it in their publication. You can manipulate this in a way. I could change the color of different things if I wanted to. Ooh, I don't even like that. I'm changing that back. And each of them, if I didn't want to see GC content, I could remove it. If I don't care at all about transporters, I can remove those. And if there are tracks I would like to add, I could add them. Like, let's say I wanted to see all the mobile element proteins. And say I want CDS on both strands, and I can add that in, and it's going to generate it on the fly, except now it's so low down and the color is bad you can't see it. Going over to sequences, this will just show me the contigs available for this genome. This is a reference genome, so it's a complete genome, meaning that the number of contigs match the number of chromosomes in this organism. And the only reason I bring this up is that this is the one place in the resource that you can get the FASTA data for the entire contig. So this is the only place you can get it. So I wanted to bring that up. Proteins. When we started this, we were at one single protein for that ESX gene. This is the number of proteins across this one individual genome. You can see that it's got 4,367. It's already scaled to look for coding sequences annotated by the BV-BRC. If I wanted to, I can open up the filter. I can change things. I could look at add in the RefSeq information, or I could take it out if I didn't want to see it. I could do other things, filter on those, or I could use a keyword filter to say, I want to see all the genes that have hypothetical in their name. And so you can do that just as quickly. Remember, for the individual gene, when we looked at protein structures, and there were like, I don't know, 8 for that individual gene. These are the protein structures available across this entire genome, which is 624. And if you wanted to see like how many protein structures are associated with this gene, perhaps. I thought there was only going to be 2. I hadn't looked, and here there are these from the different resources. And if I put in the filter, I'd be able to see what those were. Specialty genes. I talked about this earlier. If you go in and annotate your genome, or any genome that we bring into the resource, and perhaps at this time, this is a good place to say this. We bring in data from GenBank that we come in almost every day to bring in new data, and we get them from some sequencing centers, and some people make their genomic data public in our resource. But also for some things, especially antimicrobial resistance, we have a team that has been using that data for certain taxa. They go in and they read papers, and what they're looking for are large studies that are using hundreds or thousands of genomes and looking at the antimicrobial resistance. Generally, those are mentioned in papers, but those genomes are not available in GenBank because they had hundreds or thousands of genomes, so they put them in the sequence read archive. We bring that data in and we assemble and annotate it so that we can run our own machine learning algorithms to identify areas of a genome that seem to be indicative of resistance or susceptibility to different antimicrobial compounds. And so we provide that here for you, so you can look at those too. So I could open up the filter here, and I could say, I want to see BLAST-P evidence for this, and I could, I've got it set for human homolog. I've got to go back and unset that. I want it for antibiotic resistance, and I want it for, let's say I wanted to look at NARDO information from the National Database of Antimicrobial Resistance that they have here. So you can see what those genes are, what hit it when this is coming up. And I'll be able to show you the K-mers at a little bit higher level. But this is the kind of information that you can use, and you can click on it, and beyond the green bar, it tells you more information about it. Domains and motifs, we mentioned that for the individual gene. This is across this entire genome. And let's say you're interested in HNH. I'm not particularly interested in, but it's one that I remember. You might be interested in, and now I can remember it's _5. How many genes within this particular genome have this particular domain? So you can use things to focus on, to drill down your focus and look for things too. And I'll keep clicking on this so you can, because I want to point it out that this is an important functionality. Let's click on protein families. Once again, any genome annotated in our system is assigned protein families. The reason we bring this in, you may be saying, why are you taking the data from RefSeq and GenBank and re-annotating it? Why are you doing all of that? Because we want you in this resource, when we consistently annotate everything using the same pipeline, you can make direct comparisons between those. And we have several tools that rely on those particular protein families and looking at them. We have a protein family sorter tool that allows you to compare those things. The Bacterial Genome Phylogenetic Tree Service will call on the protein families to do that. Moreover, it allows you to make a direct comparison between things. So you're not talking about something annotated in one resource and annotating in another resource, and which one is which, and do I have to look at the start and stops to be able to figure out what they're called in each? This way they're all called the same thing. So if when you do the annotation service, you define at least a genus, you'll get two types of protein families. The genus specific or the local families are genus specific. The PG families are the global families, and they're cross genus. And so this is telling me these are all the protein families that are available on this genome. For this one genome, there are 4,265. If I want to, I keep using hypothetical just because I can generally spell it without having any mistakes. You could filter in on that to be able to find all of those. And not only do you have this table where you can say, oh, I want to see, looks like there are two proteins here. Maybe I want to see multiple sequence alignment for those, which you could click and that would open it up and it would show you what that looked like. But you can also have a heat map view that shows you the genome and the protein families, the individual proteins in each of those families that were identified. And this is something we'll focus in more on when we do the tools and services on April 1st. Every genome annotated in the resource, we look for EC numbers, and then we map those genes to KEGG pathways. And if you've ever used KEGG, you'll notice this looks very familiar. Here's the KEGG pathway with the same weird color that they like to use. This tells me in the EC numbers and how many genes in my genome have that. If you wanted to see more of it here, well, also you can do direct comparisons between genomes using the compare pathway view, but we'll talk about that on April 1st. For anybody who's used KEGG pathways and have tried to find individual genes, you could like go blind doing this. So we allow you to click on it and see where those guys are in the individual pathway. You can also see a heat map view of those, which isn't so interesting, I admit, when you're looking at an individual genome, but when you're looking across and comparing multiple genomes, it's really good for you to do that. And then we have subsystems, and actually this sometimes takes a while to load. So I preloaded this page so you could see this. This is an effort that is done by the Fellowship for the Interpretation of Genomes that are part of the resource. And they're biochemists and other people who are part of this, and they are linking together not just pathways, but things that are known to work together. So you could even do something like type in virulence and sometimes this is a little slow. We're working on this to make this better and faster, but then you can look at things. And what I particularly like about this, when I'm in a KEGG pathway and it loads the KEGG pathways, and it'll even have things like photosynthesis. If something has a particular EC number and it maps to the KEGG pathway for photosynthesis, we show that. But Mycobacterium tuberculosis H37 RV does not photosynthesize. So it might be confusing to someone who's not thinking about those things. What I like about this is it's telling you for this particular subsystem, this is active, or it's likely to be active. So you can look at this and it'll tell you more information, how many genes are part of the subsystem for this particular one in this genome, and how many roles. So it means that there are 15 roles, so it's like 3 or more could be for that, and it's an indication that it is active. So, and you can download this information. I play with it all the time. If you're a user of RAST, this is one of their most awesome things that they have. Experiments. We looked at experiments earlier when we were at the individual gene level. This is at the genome level. And so I could choose, oh, I want to see, let's see, maybe I'll choose this guy. Let's, just choose one I want to see. I could click on the experiment tab and it would give me the information about that particular experiment. Or I could click on the Biosets tab that allows me to manipulate that information across each of the experimental conditions that were part of that experiment. So, okay, sometimes you have to wait for things. So I can go in and I can say, oh, I want to see, it says that there are 7,377 genes that are interacting there, and I could filter it by one log fold. Ugh, still too many. Look for two. I mean, it's not that it's too many. I just want to show the next thing that's actually pretty, so I can see which are either going up or down very strongly. So this is a way to get it. You could also do the heat map view, and you look at that and you think, oh my God, I can't make any sense of that. It's too complicated. Well, that's why we provide this cluster button. And so you can click on that, and then it will reorganize the heat map view to cluster together the data of things that are either going up or down in a certain level. And you can mouse over it to see it, or you can grab it and actually say, show those genes, add them to a group, or download those genes. So that's interact. So that's experiments. And then we also have interactions. So I'll give this, sometimes it takes a little while to load. I'll give it a second here, but I don't have too much time, so I don't want to wait too long. But I could filter in and I could say, I want to see all the interactions for, let's say, direct interactions. And then I could maybe click on those and say, I want to see the graph of those. And then you can pull that in, and then it gives you some nice, a nice little visual there. So I can see mousing over those. These are the host proteins. This is the gene one of the genes I was on. And then you could even see the interaction there and what the experiment was. So we started with the genome, and we went through all the tabs, just like with the gene page. You start, there's the overview page, but don't forget about the tabs because there's so much more information there. Let's take one step up and look at the taxon page for something, and we'll just do Mycobacterium tuberculosis. So let's click on that, but I could click on any of these and we could see all that data. So for Mycobacterium tuberculosis, things look a little bit different, but you'll notice that there's, whoops, I was waiting for that to load. That's why I was saying it looks a little bit different, but it's not what I'm used to saying. It's showing, this is something new you haven't seen, the genomes by antimicrobial resistance. We have the taxon summary data. We have the reference and representative genomes associated with this particular species. And look, there are 29,696 genomes for Mycobacterium tuberculosis. The data is just getting huge. So finding the data that you want to look at to compare with your private data, that becomes more and more of an issue. So those filters, the keyword text, those are all going to be very important for you. We have ways for you to, each of these, we could go to each of these genome pages, but we can play with this. I just like playing with that little box. Each of these is a hyperlink, so I could go to this, to the strep resistant or strep susceptible. I could go to the human isolates or the ones that came from South Africa. Each of those are hyperlinks from this page that allow you to go to the data you're interested in. Phylogeny, this will be exactly the same that we saw for the genome because it's the same group. Taxonomy, kind of boring because it's just in Mycobacterium tuberculosis. So this is just a kind of boring list of the strains and genomes within this taxon. If you were at a higher level, you'd see more diversification here. But this is where things are about to get interesting. So like I said, there are 20, well, of course, it's been interesting the whole time, hasn't it? But this is where it gets even more supercharged interesting. So there are 29,696 genomes in Mycobacterium tuberculosis. And there's just nobody well, maybe some of you want to deal with all that amount of information. I don't. So to winnow things down, I would click on the filters and I might say, oh, look at this. There's false and there's true. What does that mean? Well, if it's true, it means it's a public genome that anyone can see. If it's false, up here, you can tell this cute little icon of the head and shoulders. That means I'm logged in. So this is my private data. If I were to click on the false, that will show you what Mycobacterium tuberculosis data genomes I've annotated in the resource. Now, so you can see them now, but when you're logged in, you won't see my data. I'm going to click off the false. We're just back to the true here. So it's still, oh, it's just too much data. It gives me a headache to think about it. I might want to see the complete genomes. Every genome that's annotated in the resource, we give it a quality score that's available on the genome landing page to tell you if it's good or poor quality. So I might want to see that. And let's say I want to see the genomes that have been isolated from humans. And I want to see the genomes that have been isolated from humans in Asia. Okay, now I'm down to something I can see. But now what do I do with this? It's still, you notice, it's looking across 29,696 genomes. So I could click on all of these and I could create a group of them and look at them later, or I could rescope the view by clicking on this apply button. And notice it's showing all the information of all those things I just clicked. And now it's rescoping everything dependent on that. So if I go to Amr Phenotypes, it's going to show me the data associated just with this group across these 71 genomes. And so I could go into the filters and I could say when it says add a boost classifier, that's indicative that these are computational methods. This is from our machine learning algorithms that are predicting based on the sequence of this genome if it's resistant or susceptible to a different antibiotic. And if I were to click on the genome name, you notice that these all have the same name. It resorted, sorry, I didn't explain that. Any of these column headers is sortable. So if I click on it, it sorts it. And the reason I wanted to show you that is that this particular genome is predicted based on our machine learning algorithms to be susceptible to this and this, resistant to this and that. And then later, when I drill down more deeply into the individual tabs on other things, I'll show you how you can visualize that information, but that's pretty cool. Sequences, I don't want to click on that. It's just going to show you the contigs for this particular group. Proteins would show you the number of proteins for this thing that I've scoped it to. protein structures, might click on that. Because one thing you might want to see is, you might want to do the HNH. Was that the guy I was looking at before? I think so. Well, probably not, because I got that wrong. But anyhow, so you can look, probably because none of them have that particular structure within this group, which makes sense. Specialty genes. Let's take a quick look at that. And once again, and you guys are probably going to be saying this in your sleep, if you annotate a genome in BV-BRC, every gene is blasted against these specialty databases. And so let's say within this group, I was interested in the virulence factors, and I was interested in the virulence factors that were part of this curation attempt that we did at one point. And let's say maybe this is the one I'm interested in. And you notice it gives you a lot of information beyond the green bar, telling you everything you possibly wanted to know about this particular virulence factor. So things like that, this is that kind of information that we provide. Domains and motifs are across just this group, so I'm not going to drill into that because I wouldn't say I've bored you with it before, but I've talked about it before. You might say that I bored you with it before, but hopefully not in front of me because it'll hurt my feelings. Epitopes. This is something new. You didn't see this before when I was at H37RV, right? So this is something new that came from our interaction with JCVI and IRD. This is something that they provide. It's being a little slow for me right now, but you may be, well, I mean, when it came up, I was thinking, what is an epitope? I can't really remember. Well, the epitope, the antibodies that are floating through your body, when they find something antigenic and they bind to it, the part that they're binding to is the epitope. And so this is showing you the epitopes associated with this group. This is brand new. So you bacterial people coming from Patrick, you could tip your hat to JCVI or the people from IRD and Viper, because they're the people who originally brought you that, and we've brought it in for bacteria too. So when I want to look at one, I can click on it. It populates information beyond the green bar. I could filter in on things if I wanted to. Pathways, subsystems, experiments, interactions, all of those I've gone through before, so I don't want to go through those again right now. But so we've done the gene, we've done the genome, and we've done the taxon level. But all of this, I mean, I talked a little bit about your private data, but and I'll show you more of that in a second. But what if it's an organism? I can't tell you, this happens almost every week with me. Someone says to me, I can't use your resource because you only do bacterial pathogens. You don't have the information that I'm for the organism I'm interested in. Well, you could click on all bacteria, which would show you, this is of course at the bacterial level, but it just gives you an idea of the number of genomes, of the number of bacterial genomes that we have in BV-BRC. But even that, let's say there's an organism I'm interested in. Oh, how do I find it in this? I could go to the genomes page and try to look for it, or I could, I could look for it using the global search. And this is one of my favorite organisms, which is an endosymbiont of the tsetse fly. And so I can, just using global search, I can find it, and let's look at it. And it's showing me that there are 4 genomes in BV-BRC. If I click on the filters, I can see that there are 2 true genomes, which you probably, if you remember, that means they're public, and 2 false genomes, which means that they're private, not that they don't really exist. It just means the private. And I know I don't get the true false part either. but the computer scientists, it's important to them, so I don't fight with them anymore about that. So let's click on the true here to see the true ones. And so this is not a pathogen, right? This is something that's important to this tsetse fly. So what's it going to look like when this time I click on this genome landing page? Is it going to be totally different? So let's click on that. And no, everything looks the same, right? We're seeing the same page with all the same tabs there. So you can see that. So let me go back here. Now I want to see if it's my private data. I know you're probably thinking, yeah, we got it. You don't have to beat us to death with this thing. Hold on a second. There we go. So I could click on the genome here and look, this is my private data and it looks exactly the same. So I just want to emphasize to you, we try to make, we structure these things so that it doesn't look radically different depending upon the feature, the genome, or the taxonomic level, your private, your public data. All of this should look, we want you to be able to step through it and see it. And I think that should conclude everything that I've